Swayam Prabha. Digital India. Educated India.
I'm Swati Tiwari. I'm a faculty at the School of Biotechnology, Jawaharlal Nehru University, Delhi. And this project was supported by DTH Swayam Prabha, MHRD. Okay, so now we come to the last lecture of the chapter on vectors. And in this lecture, we are going to discuss about what are the various vectors that are used for animal cells. So we will first discuss what are the essential features of vectors that have to be used for animal cells. Then we'll talk about some of the very basic strategies that are used with viral vectors that are employed in animal cells. And finally, we will take an example of a very popular plasmid vector for mammalian cells, which is known as pCDNA3, and we will explain what are the various parts of this plasmid vectors, and this is basically similar features will be observed in most of the other plasmid vectors for animal cells. Okay, so the first thing is that we need to understand why we really need vectors for animal cells. So for biotechnological applications, mostly when scientists need to produce a recombinant protein, they express it in a heterologous system. For example, E. coli is the choice vehicle to be used as factory for production of heterologous proteins. Or in some cases, yeast is also used, but yeast is mostly used to understand the genetics and to find out the genes which are responsible for regulating certain functions, etc. But the problem with the prokaryotic or even yeast system is that sometimes when you express animal proteins in this system, then the protein is not expressed correctly. So what are the reasons behind this is that in higher eukaryotes, we know that they have organelles and many membrane and secretory proteins. They are post-translationally modified. So those modification systems which are provided by the endoplasmic reticulum and Golgi in the higher eukaryotes or in even early eukaryotes like yeast, they are absent in E. coli. And even though yeast has the post-translational mechanisms, they are quite different in the extent of glycosylation that can be achieved, or even some other modifications that are put on the proteins in case of animal cells, and they cannot be done in yeast. Because of these reasons, sometimes the animal cell proteins, they are not produced correctly in E. coli or in yeast if expressed in them. So obviously then specialized vectors are required which can introduce the gene of interest in animal cells and produce the correct proteins. Now the complication there is that just like in plants, that every plant is different. So monocotyledonous plants and dicotyledonous plants. So you need special tricks to be able to express and even introduce the gene of interest in those plants, as I discussed in the last lecture. So similarly, for different eukaryotes, you will need specialized vectors. So for example, if somebody is working on C. elegans, versus somebody working on zebrafish or mouse, they will all need different types of vectors which will work in these different organisms or cell lines which are derived from these organisms. So, because in animals, most of the attention has been given to mouse and human cells, and because there is medical interest to be able to generate DNA vaccines or even other types of gene therapies. So, because of that, most of the attention has been directed at cloning systems for mammals. So before we go into what are the various cloning vectors for animal cells, first let's understand how the eukaryotic genes organization and their expression is different from that in prokaryotes. So we all know that in prokaryotic genes, we have operons. I explained to you the lac operon earlier, where several structural genes which are required for a specific film, they are governed from a single promoter. So these in prokaryotes, the genes are polycistronic. And they do not contain any introns. Now, introns are the non-coding sequences that are found in between the coding sequences in eukaryotic genes. So, eukaryotic genes, they contain introns, which are the non-coding sequences, and they have exons, which are the coding sequences. So, the transcript that is produced from the eukaryotic genes, it will have both the exons and the introns. The introns are not required and they will not allow the translation of the complete protein, so they need to be removed. So they are spliced out to generate a full messenger RNA for coding for a protein. Another difference between prokaryotic and eukaryotic transcription is that in eukaryotic systems, the messenger RNA, after it is made, it acquires a 5' cap, which is basically a methylated G residue. And this methylated G residue forms a 5' to 5' triphosphate linkage with the next residue with which the mRNA was initially started. So basically, you remember that in DNA, the linkage is 5' to 3' phosphodiester bonds. But here, this is a special 5' to 5' triphosphate linkage of this methylated G residue. And this is added on the mRNA 5' N. And this basically protects the mRNA from degradation. Additionally, at the 3' end of the eukaryotic mRNA, there is a long tail which is basically all the A residues. So it is a polyadenylated or poly-A tail. And this sequence is not actually present in the gene, but it is added after the RNA has been transcribed in the nucleus. So this is showing the same thing as a picture. So if this is the DNA and this particular gene has 3 exons, exon 1, 2 and 3. There is a promoter, and of course there are many other regulatory sequences which I am not showing here. But in between these exons, these regions which are shown as intron, these are the ones which do not code for the protein. These are non-coding sequences. At the end, there is a signal sequence for addition of poly-A tail, and then there is a termination signal. So first, a pre-mRNA is formed, where all these exons and introns, they are all press pre-mRNA along with the signal for addition of the poly-A tail. Then these untranslated regions or these intronic regions, they are spliced out, they are removed, and then the final mRNA is produced, to which there is a 5' cap is added with the methylated G residues connected via 5' to 5' trisphate linkage, and at the 3' end a poly-A tail is added. So now you see that the eukaryotic transcription is much more complicated. So for the expression of eukaryotic genes, generally people isolate the RNA and then they make a complementary DNA and use that cDNA to clone the gene into a cloning vector. So what are the essential features of the animal vectors? So basically the vector should be a shuttle vector, because most of the cloning part, as we saw also in the plant vectors, that one should be able to do it in E. coli, and the reason it is done in E. coli is because E. coli is easy to handle. You can grow it on a very cheap medium, so it's not expensive. You can also grow it in large quantities to be able to isolate a lot of DNA, or plasmid DNA, that can then be used for all the experiments. So E. coli is the popular choice and for expression of or cloning of any gene. So if you have a shuttle vector which has the origin and selection markers for both E. coli, where all the work can be done initially, and then isolate the plasmid and use it in the animal cells. So it should also have the origin of replication and selection markers, so that one can select out the clones of animal cells which have received the cloning vector. Also, because the difference in the transcriptional machinery of the prokaryotes and eukaryotes, so the promoters should be such promoters that can be recognized by animal cell RNA polymerase, and therefore the expression can be achieved. And in addition, these vectors should also have the sequences for termination, polyadenylation, transcript. Other than these features, there are a couple of other sequences which are required for mammalian cells. For example, it is known as a Kozak sequence, which is required at the 5' end for efficient production of the protein from the animal cells. So, to achieve all these things, defective animal cell viruses have been developed as vectors, and these animal viruses, they work on the principle that the defective function is provided by the host cells, which have some of the viral genes that is integrated into their genome, and therefore the missing function of the virus is provided in trans, just like plasmid vectors we talked about alpha complementation. Similar idea is used here, that you have a defective virus which cannot really replicate, and then that replication function is provided by the host cell in trans, and these vectors have been engineered by deriving sequences from animal cell viruses, just like bacteriophages were used to engineer vectors for E. coli. Similar way, the animal viruses have been used to devise these vectors for animal cells. Okay, so the first viral vector for animal cells was engineered from SV40, which stands for simian virus 40, way back in 1979. And SV40 is a polyomavirus, it's a DNA virus, and subsequently many other vectors have now been used as vectors. So, for example, papillomaviruses, adenoviruses, baculoviruses for insect cells, lentiviruses, and many such viruses have been developed as vectors for animal cells. So let's see what is the SV40 based vectors. So as the name suggests, it's a simian virus, meaning that it is a monkey virus, and it causes latent infections. What that means is that its genome, it gets integrated with the host cell chromosome, and it has a circular double-stranded DNA genome, and it's a small genome of 5.2 kb. So it is just like a bacteriophage, M13-like size, and has 6.4 kb genome, and similar this one has a 5.2. So it is very easy to handle, and one can use it as a vector. And it encodes 5 proteins. So this genome of this virus has 5 proteins encoded in it. One is a small T antigen, and then there is a large T antigen. And this large T antigen is very essential for viral replication. Then there are 3 virion proteins, VP1, VP2, and VP3. So these are required to make the capsid of the virus. And of course, it will have its own origin of replication, which is about 80 base pair long. So the early vectors, which were based on SV40, they contain the origin of replication and the VP1 to 3, these virion proteins. And these vectors were used in a cell line, which was a monkey cell line known as COS. So there is COS1, COS7, and several such cell lines which have been derived from African green monkey, and they can be grown in culture indefinitely. So this is a cell line in which the large T antigen is integrated into the genome of this cell line. So the cell line can be used with these SV40 based vectors because this vector does not have the large T antigen, but it does have the origin of replication. So the large T function is provided from the host cell genome, and therefore in this cell line, this vector can replicate, but it cannot really get packaged properly into virions. So it produces a very high copy number per cell, and the copy number is not replicable, and therefore the cell has a lot of metabolic stress on it in order to produce so much of your protein from this vector in which you have cloned your gene of interest. So the host cell becomes inviable. So the later vectors were known as late replacement vectors, and these basically again had the origin of replication, and along with it they also had the splicing and polyadenylation signals, as well as the entire early region of the SV40 genome. And the region in the genome which contained the VP1, VP2, and VP3 virion proteins, it can be replaced by the DNA insert of your choice. And these vectors were used to infect the host cells along with the helper virus, which produce the virion proteins for the packaging. So now the virus this viral vector can basically replicate, and the gene that has been can be expressed from this because it has the splicing and polyadenylation sequences, and because the virion proteins are produced by the helper virus in the genome of the from the host, not from the genome, I'm sorry, from the host cell. Therefore, the cells that are infected by both these viruses, the vector virus as well as the helper virus, they can produce virions. So you can isolate a large quantity of these virions from these cells and then use it to infect the cell line of your choice in order to produce the protein of your interest. Alright, so other than SV40, many other viruses have been developed as vectors, and prominent among them are the adenoviral expression system, the adeno-associated virus system, of course, retroviruses and herpes viruses. So, in general, all of them, they use two plasmid vectors, and the viral vectors, when you express a gene of interest from these viral vectors, they can be of two types. In one case, there can be an integration of the gene of interest with the host genome, and this happens if you're using viral vectors and adeno-associated viruses. And in other cases like adenoviruses and herpes virus vectors, the gene of interest does not integrate with the host genome. So, except for retroviruses, in general, two plasmid vectors are used. One is a cloning vector, which is obviously a shuttle vector, and second is a vector, which also again is a shuttle vector, but it has the entire virus backbone, and that viral backbone is missing certain regions, so certain genes are deleted, which are required to replicate the virus. So these deletions, because of these deletions, this viral backbone will generate a virus which is a replication incompetent once it is combined with the rest of the sequence that is coming from the cloning vector. So in many cases, these two vectors, they are introduced in a packaging cell line, either directly or first they are introduced in E. coli for recombining regions of this cloning vector with the viral backbone, and then introduced into a packaging cell line. So this packaging cell line is a cell line that will provide the missing genes from the viral backbone, and this will allow the replication of the viral genome in this packaging cell line. So because these functions are being provided in trans, therefore, even though the virus can replicate in this particular packaging cell line, once this virus is used in another cell line for infection, then in that particular cell line, this virus cannot replicate because it's a replication incompetent virus. It is missing those genes which were required for its replication, and because those gene functions was being provided for the packaging cell line, where those genes are integrated into the genome of this packaging cell line, these functions are not available in other animal cells in which you might want to infect this virus. So the virus will infect those cells and produce protein, but cannot really multiply in those other cell lines. So this replication incompetent virus with the gene of interest is used to infect the animal cells, and the gene of interest can then be expressed like a viral protein. And how much expression of the protein is achieved will depend in part on the multiplicity of infection. And the multiplicity of infection basically means how many viral particles per cells are there, and higher the number of virus cells, higher the amount of expression. Now the problem is with these viral vectors in general is that it's a very laborious and long process, because first you have to clone the gene of interest, then you have to regenerate a viral backbone in most other cases. For retroviruses it is simpler process, but most of the viruses, it is a long and laborious process. And also there is a danger always present that the wild type virus can be regenerated if there is some recombination between the genes which are integrated into the packaging cell line and the viral backbone that you are putting in that packaging cell line. So if there is some recombination, a wild type virus can be regenerated. And so there are many versions of these viral vectors that have been produced, because viral vectors have been explored as a gene therapy vehicle. So one does not want to reproduce or regenerate a wild type virus in those cases. So there are many generations of these vectors now, generation 1, generation 2, generation 3, and basically all the effort has been to make the system and develop the system in such a way that the possibility of regeneration of wild type virus becomes almost negligible. So because of these problems, in most of the cases in general laboratory methods utilize plasmid vectors. So now we are going to discuss one of the most popular plasmid vectors that is used for mammalian cells, and this vector is known as pcDNA3. So this is a plasmid double-stranded closed circular DNA molecule of 5.4 kilobases. Now if you see this map here, you can see there are many things that are marked. So first let's try to understand what are these different parts of the plasmid, because most of the plasmid vectors for animal cells you will see, and even for bacterial plasmids, you will see many such things which are marked. So let's try and understand how to really read a plasmid, because that is very important to understand which particular plasmid you can use in which particular cell type. Okay, so this is a mammalian plasmid expression vector. So first of all, I told you that one essential feature is that it should be a shuttle vector. So it should have origin and selection marker for E. coli. So you can see there is col E1, and col E1 is origin of replication for E. coli cells. Then it has ampicillin selection marker, which is shown here. So this is a selection marker. When you code your gene of interest, then the recombinants can be detected based on ampicillin resistance. And then one has to have a promoter that can function well in the eukaryotic or animal cells. So this promoter CMV, PCMV, stands for promoter derived from cytomegalovirus, HCMV, human cytomegalovirus. And this promoter is a strong promoter that can drive transcription of the gene interest that you will be cloning in the polylinker region. Now here is the polylinker region, but you see that there are two other sequences shown here in opposite orientations, the T7 and sp6. Now T7 and sp6, these are promoters which are derived from bacteriophages, and these are again very strong promoters, and these promoters are introduced here on both sides of the multiple cloning site, so that sometimes people want to do in vitro transcription or coupled in vitro transcription and translation of the gene of interest. So for that, purified T7 polymerase or sp6 polymerase can be used to drive the transcription and translation of the gene of interest, which is cloned in this polylinker region. Now beyond that, you see this BGHPA. Now BGHPA stands for bovine growth hormone polyadenylation signal, And this is required to terminate the transcription and for the polyadenylation of the mRNA that is produced from the transcription of this particular vector. Then you also see there is F1 origin, and this F1 origin is basically the origin of replication from F1 phage. And this is a leftover sequence not really required for expression in mammalian cells, but as in case of M13, we saw that those plasmid vectors will be useful for producing single-stranded DNA. And so in case some researchers, they want to produce single-stranded DNA, then even pcDNA3 can be used, and this F1 origin of replication will allow production of ssDNA synthesis. Further, there are more sequences here. There is also an SV40 origin of replication and also product from the SV40, and this can drive the transcription in cells which express the large T antigen from SV40. So there are many animal cell lines which use which have integrated large T antigen in their genomes. So those in all those cell lines which have this large T antigen, you can use this plasmid for expression. So human embryonic kidney epithelial cells, which are known as 293 T cells, these can be transfected using this particular plasmid. And also, because there is a neomycin resistance gene here, if you can actually select the transformed cells, so this is the mark which is used for selection in animal cells. And then again you have an SP40 polyadenylation sequence for polyadenylation of this neomycin resistance gene product, mRNA. And therefore, if you have a long-term selection for this neomycin, then stable transfectant in animal cells can also be generated using this particular plasmid. So to summarize, I have told you that animal cells, they require specialized vectors for gene expression, and you need specialized vectors because there is a requirement of recognition of promoters by animal cell RNA polymerase, and also for polyadenylation of the mRNA that is produced. Many of these animal cell vectors, they have been derived from animal viruses, and there are a number of these viruses which have been developed and engineered as vectors for animal cells. In general, 2 plasmid shuttle vectors are used and for cloning the gene of interest and then recombining them to create a replication incompetent viral backbone. And in most of the cases, it is done in E. coli, but in case of retroviruses, that is not necessary. And this replication incompetent recombinant virus is generated in a packaging cell line, which produces a replication incompetent recombinant virus, and then that virus is isolated and can be used to transduce a target cell line to express the protein of interest. But because the viral vectors are more laborious to produce and it requires time and experience to use them, therefore a much easier system for expression of animal cell protein is to use the plasmid vectors, and these are again shuttle vectors which have sequences which are basically derived from animal viruses. So we have promoters derived from animal viruses, and also the polyadenylation sequences are added to these plasmid vectors so that the host cell will allow the replication replication, transcription, and translation of the clone gene of interest. So this brings me to the end of this entire chapter on cloning vectors for prokaryotes and eukaryotes. I hope that many of the concepts are now clear to you, because we will be using these concepts later on in many chapters that I'll be discussing later on. Thank you very much for your attention.